So in this field trip, we went to Nagasaki Prefecture Institute of Environmental and Public Health. This institute focus on research on environmental problems such as toxicity, water quality, protection, and propagation of aquatic species. The public health department also focus on such as topic COVID-19 and the spread of E. coli. Then after that, we went to Institute for East China Sea Research. This institute focus on research toward fisheries resources and aquatic habitat. They also focus on experiment toward local species to understand more about the biology of the species. The main objective of this institute also focus on the protection and restoration of the marine environment and ecosystem by investigating the fundamental mechanism that drive material flow and bioproductivity in the marine. This institute also unraveling the mechanism that will ensure a sustainable use of marine bioresources by understanding how the dynamic of the environment affect reproduction in marine organism. So this is the laboratory attachment from 10 June until 21 June where we went to lab Sakakura Yoshitaka, and in this lab there were many activities that has been carried out, which were initial culture of rotifer and copypot. So in this section, we inoculate the rotifer culture with batch culture. And for the rotifer, we want to observe the reproduction rate of the rotifer, and for the copypot, we want to observe the morphology change of the female. Next, we went to inoculation and harvesting of microalgae, where we inoculate the Nanochlorosis oculata, and we harvest from the previous culture. So the process start with harvest the previous microalgae and filter 100 ml and transfer them into 900 ml of natural seawater, and then we inoculate in the microalgae culture room. So this is the process where we harvest the microalgae before we can feed to the zooplankton. Next is the filtration of natural seawater and preparation of 22 ppt of seawater. We use the Iwaki aspirator 13 to filter the natural seawater, and after that we use the seawater to prepare the 22 ppt of seawater, because in this lab microalgae were cultured at 22 ppt of seawater. So next is the Professor Kim and Satuito laboratory, where the first activity was we inoculate the Nanochlorosis oculata. So in this process, it's like same with Professor Sakakura's lab. Then we continue with initial culture of rotifer. So for Professor Sakakura's lab, we culture in batch culture, and for this lab we culture in individual culture, where we also want to observe the reproduction rate of rotifer. So next is the microplastic observation in rotifer. So this is the microplastic under microscope, and then we dilute the microplastic into culture media of rotifer, and we observe the digestive tract of rotifer and also their faces. So thank you so much for watching. Bye bye.